Hi, once I showed you this title and promised that I will explain later what Glimma is about, and by this time I hope that you know already what is Glimma and HR. And you can also check a bioconductor page for differential expression keyword and find that Glimma, HR and dissect are three and only packages which do differential expression analysis, Glimma for microarrays, HR and dissect for bulk RNA-seq data. And if you scroll a little down, then you see that Glimma is also mentioned here, which is interactive visualizations for gene expression. The idea of interactive visualization is probably clear, although the benefit of it, especially for this case, is less obvious. But we can always have a closer look. And when you study the new package, there are usually three steps. First, you read the instruction, user's guide. Then you check the examples provided by the user's guide. And then after it is clear how to use it and with what kind of a data, then you try it with real data. And then we will see what the extra value of interactive figures will be. The package is small, but remarkably it has three users guides, one for application in Dissect2, another is for Glimma OHR, and the third one is for single cells with. If you go to the reference manual, and reference manual is a list of all functions in a package, you will see that it's basically Glimma MI plot, Glimma MDS plot, Glimma Volcano plot, and general use XY plot. That's all it is. And GL is the HTML version, whereas Glimma version gives an output within RStudio. If you click on HR button, then you will see lines with the example dataset which you can copy paste in your RStudio, and then right after importing this dataset you can immediately do the MDS plot, which is interactive indeed. Even in the tutorial you can do colors, or you can change coordinates. Dimension 1 and 2 are defaults, but you can easily do number 3 for instance. And then you can do further analysis and either following womb scenario or GLM fit scenario, and after you did it, then you are ready to do MA plot, which is this one, and again it is interactive. And by clicking, first you do sorting by p-value, and then clicking any gene out of the list, you will see that this, for instance, appears on the plot where it is in a format of average expression, which is quite high for this gene, and log fold change. And then you see the difference in expression between those groups. And pay attention that in case of HR, contrast is explicitly added, so you are never confused by what you compared with what. And similarly you can do volcano plot, and again first you sort by p-value, then you say something like give me this. Then on the right side you will see the differences in expression between groups, and here you will see a position of actually the same gene. And likewise, if you open this page, then again you get instruction that you load example dataset, then declare the variable DDS, where you declare everything, count, samples, and design. In addition, they also suggest to add genes descriptions over here. Okay. Then immediately after this you can do MDS plot. Sure you can, and it's interactive again exactly the same way. Then you do the famous dissect line, which analyzes everything. Quiet is true is optional if you don't want to see the steps while executing this line. And then you call MA, which is equally interactive, only now you don't see gene names, it's entries numbers. So, and then you can move on, and they don't show volcano plot, but it should be possible as well. And they don't tell much about web versions of these figures. You can copy paste all the lines from the user's guides into your RStudio, and then you have to add a few more lines like declaring libraries. Then for first time you can inspect what is actually your data source, what is DGA exactly when you import it, and then you see that it has a format already compatible with the HR or dissect data analysis. It is equivalent of the line DGA list declaration in HR or dissect data set from matrix line in dissect2 protocol. And since you know what the data are, then you can place your figures. The MDS plot comes right after the import of the data, obviously, and then it comes not into the usual bottom right corner, but in a special window studio viewer. It has exactly the same features as you have seen in a tutorial. The only extra exercise can be using web versions of the figures, and similarly you position figures for dissect protocol as well and test whether you can get all those figures. But the real test is of course with the real data. Let's have a look how it will work with the real data. I decided to take a GLM HR version of HR, which is script number 5. Details can be found in this video, and the script itself is also on my GitHub, which is this one. We load what we need, then load the files through the same read DGA and make the table, then omit NA. So it is a little bit longer business. I go very briefly. So now we need the sample information to figure out what is what. Then we reduce the table and add the categories young and old. So the major result of this part is a reduced table which describe the samples. Alright, now it's time to declare DGA list variable. Then we do necessary step of filtering. So it's basically a good data set. And we do either regular MDS plot within, or we do a Glimma MDS like this, by age again. This is the quantitative age from low, and we need young and old groups. Now we divide it. Okay, looking good. Or we can do it HTML version. Only here we have to declare groups in advance. Then again, it is going well, and label is over here and groups. Now we do differential expression analysis and estimate dispersion and plot BCV and dispersion coefficients. After this we do fit and design, and then we pick groups, what we compare with what, and we say group old versus group young. The result for defined contrast, GLMRT, and then MD plot within HR script, or MA in a studio viewer, old versus young, interactive. It's all working well. Yeah, this one for instance, or we adjust and try again. There is a bit of a problem that at this step there is not yet an annotation of the genes, so we operate only with ensemble gene numbers. For the rest, that's okay. And we try also HTML. Here we missed colors, so it's less fortunate. And then we test Volcano. Volcano plot works as well. Okay. The rest is not so much important for our exercise that's getting into the annotation and tops. Instead, we check how it works in dissect2 with the real data. I selected a data set with the 3 groups and 2 factors. And of course, I explained this script before. More details are in this video. Here I will go through pretty fast. First we import what we need, then get gene annotations, then geo is information about samples which you can always get online, and get the sample information and reduce it. On the basis of this information I simply generate 3 groups, 1, 2 and 3. These 2 are controls to this major group, and later you will see that the preferable contrast is a group number 2 versus group number 3 versus this one. Note the title column shows also information about batches. It looks like the experiment was repeated 3 times in 3 different batches, 1, 2 and 3. And we can obviously use it for our analysis. So this is the sample table which will be used for dissect control, CM control versus CMCC group. Now I do filtering. Then we can do a palette and a box plot. Alright, and also do a legend. So the first group is a bit defective, that's why I try to stay away from it. Now we go here. And now immediately after declaring a dissect dataset, we can do a MDS plot by group or by batch, which is the second factor. It works. Then online option. At least in this set it works. How I change it by batch I don't know. This part is not relevant. We go to PCA, to a heat map, SD plot, I skip it all. Now dissect works in a 2-step. First you do a dissect analysis either with VALT or LRT test type. And now you ask for Volcano, and now you feel a little bit confused because it's only one differentially expressed gene, and then the question is what exactly you comparing. In order to figure it out, you simply call the default results and ask what do you compare here, and then you see that you compare batch 3 versus batch 1. Why? Because that's how default will work if you define design like this. So if you want to go back to groups only, then you exclude batch, and you have to rerun it with the groups only. Then you skip the middle part with all those extra illustrations, and then you run dissect again. And then you ask for Volcano, and then it's nice Volcano with lots of down and up regulated genes. There is no gene name yet because annotations come later in a dissect protocol. Here you operate only with entries numbers, but the rest works. And then likewise, if you wonder what you actually compared with what, you ask for default results, and then you see that the last group is compared to the first group. CMCC is the last one. CMCC is the last group, and control is the first. Again, it operates with the defaults. And if you wonder how I can compare group 2 with group 3, then I don't see any clear instructions. Try to figure out yourself. And MA plot works, but also it works with the default groups 1 and 3. Why? Because the structure of the DSEC2 protocol is such that after DSEC you go supposedly to the line results, where you specify what you compare, and then you can say this and also ask for R and the summary of R, and you can do visualization by other means but not by Glimma. They do not work with the results. To summarize, this package is good for the presentations, for seminars, where you can show your favorite genes on the screen and show the interactive options. But it's not that good for publications, because again, publications require static formats. If you go to saving options, it offers you HTML. You can always save HTML page as PDF, but then the whole sense of interactivity is disappearing. It is nice, good looking. It works well within HR script. It has some obvious issues within DSEC2. I haven't seen clear instructions how to deal with the selection of groups. But in combination with HR, it works very well. No major issues found. That's it.